So another very important notion that we can get out of the entire genome comparisons is the synteny. So the synteny is defined as the overall conservation of the order in chromosomes between different genomes. And this order could be maintained for genes or entire blocks of sequencing containing these genes. So it is evaluated in whole genomes, and these blocks, as I told you, could include even large portions of the genome. So in fact, this is due to the fact that a recombination or a crossing over could affect groups of adjacent genes in a chromosome, and this helps define this group of genes being on the same chromosome, this is what we call a linkage group. So here you have an example of synteny between two different genomes. Here the human genome and a fish genome, which is the tetraodon. So in order to observe synteny, what we do is generally we color code the synthenic regions between two genomes. So in order to do that, we color one of the genomes, and then we see how these different blocks, the different blocks of colors, or the different portions of the chromosome are distributed along the other chromosomes of the second species. So if we take a very basic example here of the chromosome 15 of the human genome that has been color-coded in yellow, then you can easily see that large blocks of this chromosome 15 are maintained in chromosome 5 and chromosome 13 of that fish. So this is a way to use these synthenic blocks or linkage groups in order to infer the evolutionary relationship between two genomes or two organisms. And in the case of the tetraodon and the human genome, this allowed the researchers that did this to use comparative genomics to draw a hypothesized model or evolution of these organisms. And here, in fact, the model is starting with an ancestral bony fish having 12 chromosomes that are indicated here. And you have different evolutionary paths that led to the human chromosome here and the tetraodon, the human organism and genome here, or the tetraodon here. So, based on individual gene comparison now, we can have information about the homology between two sequences or two genes. So, two genes are homologs if they have a common ancestor. So, imagine that you have here that common ancestor and that a gene duplication event happened. These two genes would follow different evolutionary paths. If a sequence would originate, would evolve as a consequence of speciation as genes in other species, so as it is the case here, then this would be called orthologs. So orthologs are genes that are appearing as a consequence of the appearance of different species. So this is what we call speciation. And if this is an event that is happening through duplication, then this would be paralogs, and this is exemplified in here. So these are very basic and important notions to have, because these are at the basis of the gene comparisons. Orthology is at the base also of this comparison, and it takes into account the fact that we need to find these orthologs using, for example, the BLAST, and you have now possible predictions for several species in AutoMCL, for example, that you could use as a database. You also have an ensemble Comparative, which is a pre-computed data for orthology and paralogy predictions based on phylogenies. You also have another database that is called EGNOG, which is evolutionary genealogy of genes with non-supervised orthologous groups. So these are orthologous groups of proteins across different taxonomic levels. So you can query these kind of orthology relationship or orthologous relationships between genes through different kind of databases. These are only examples of these. So inferring annotation is an important step based on all these alignments and all these orthology or homologous relationships between the genes. And this is done through the Gene Ontology Consortium. So if you remember during the first sessions or the first classrooms, we were talking about gene ontologies. This is basically a way to characterize a gene based on the biological process it is involved in, the molecular function it has, or the cellular component it is acting in. So a gene ontology database would contain different tools to retrieve this information, and the whole information is here shown as a tree, starting from the less precise one, we would say, to the most precise one. And this is, really associated to a controlled vocabulary that would allow you to retrieve and to navigate through these different processes, functions, and components. So each process, function, or component would help you characterize your own gene of interest. And this could be inferred through the annotations based on these sequence comparisons. So now, once you have predicted the function of a gene, you could also have mutations associated to a gene, and you would like to know what is the impact of such mutations on a function. You could also do that through databases and resources. So if we take the example of single point mutations like SNPs, you have polyphen2 or SIFT or VEP, for example, through the ensemble, that would allow you to evaluate what impact one specific mutation would have on the function of the gene. So it could be that the gene is that the mutation is introducing a frame shift, for example, or a stop that could be gained through that mutation, and all these kind of different events could be followed using these kind of databases. We will be seeing examples of this in the assignment also. We could also know the impact of the mutation on the structure in terms of gene portions that are completely gained or lost, and this could also be done for non-coding portions. So the same effect of mutations could be happening in a non-coding portion and could be affecting the following portion that might be encoding for proteins or for non-coding RNA. So examples of browsers through which you could submit your sequences or browse through pre-computed alignments are the Vista browser, which allows you to have a collection of resources for comparative genomics. And these can be used to analyze pre-computed alignments, which is really useful because it could allow you to browse the information that is available through these different databases very, very easily. You have different options. You can query sequences against multi-species sequences through the MVista. You can identify regulatory transcription factor binding sites through the RVista. You can query sequences versus whole genome assemblies through the GVista, etc., etc. You have a lot of them. And its Vista tools are always increasing, and you have a new tool that is called the RViewer, which allow you to have a way to view regions and compare them to genomic intervals. So another browser would be the Ensemble browser, for which you have different kind of resources available. This would allow you to have comparative analysis at the genome and the gene levels. You can also use sequences so portions of genomes that could be compared using pairwise or multiple whole genome alignments. This could help you to determine synteny, sequence conservation scores, gene homology relationships, etc., etc. You have here just an example of what you can do through the sequences, and this is what we will be seeing also through the alignment, alignment of query sequence using this browser during the assignment. So the take-home messages, and I will be finishing with this slide, is that you have different input that are possible. So you can query a specific portion of a genome or a whole genome. The output that is expected is to have insights into the homology or the similarity or the evolutionary distance between two features, two sequences or two genomes or two organisms. This is done through the resources that are available now for alignments. These are different resources and different algorithms that are developed for whole genome, for multiple genomes, for multiple sequences, or for global or local sequence alignment. So make sure that you understand these notions. I know I was a bit fast in explaining them, but I wanted you to have the whole picture of the essential terms that you would be encountering each time you would be opening these kind of browsers to retrieve information about a sequence. So I would really like you to take some time to go deeper into the details of each one of them, or at least the ones that are the most interesting ones for you. I've been highlighting mainly the BLAST and the CLUSTAL methods for alignment because I believe these are the ones that you might be encountering the more when dealing with this kind of sequence alignment. And don't forget that you need to understand what are the inputs that files that are required and what are the output files, because based on these output files you would know how to interpret your alignment and how to evaluate the phylogenetic distances between your sequences or your organisms of interest. So phylogeny would be, as I told you, a completely different field. I hope you got at least the most out of it in terms of algorithms, in terms of also the different notions, the different definitions. What is a global and a local alignment? What are the differences between them? What is a pairwise sequencing alignment or a multiple sequence alignment? And I hope you enjoyed the course and that you got enough information that would at least allow you to know where to retrieve now the informations that would be the most interesting for you for querying sequences. And we will be seeing some more through the assignment. I thank you all for your attention, and I hope you would be able now to play with your datasets as you wished.